I'm Sho SHIBATA from Tokyo. Today, I'd like to talk about the role of basophil in the pathogenesis of COPD. Chronic obstructive pulmonary disease, COPD, is characterized by emphysema and airway obstruction, mainly caused by cigarette smoke. And in the patient with emphysema, alveolar damage over time, and alveoli weaken and rupture. Reduced in surface area and airflow limitation cause a symptom of dyspnea, cough, and sputum. COPD has been generally considered as a non-T2 type lung disorder, and cellular and molecular mechanism remains unknown compared to that of allergic asthma. Basophils are the least abundant granulocytes, representing less than 1% of peripheral blood leukocytes. The existence of basophils in the blood as cells processing basophilic granule was first described by Paul L. Leahy in 1879, much earlier than the discovery of T cell and B cells. Nevertheless, the functional significance of basophils remains uncertain until recently. Basophils contribute beneficial to protective immunity, particularly against parasite infection. On the other hand, basophils are involved in the development of various disorders, including allergy and autoimmune disease. Basophils interact with other immune cells and non-hematopoietic cells through basophil-derived factor IL-4, IL-6, and IL-13, and protease contributing to the regulation of immune and allergic responses. I'd like to start by talking about the role of MMP12 in elastase-induced mouse model of COPD. In elastase-induced COPD model, installation of single dose of Wilson pancreatic elastase (PPE) to biopsy mice result in the substantial cell infiltration at days 3 to 7, and followed by the enlargement of lung air spaces at day 12-21. Gene expression of MMP12 in the lung, we showed here, was significantly upregulated at day 3 and day 5. Then, intragastric administration of selective MMP12 inhibitor, MMP408, from day 2 to 9 suppress the formation of emphysema. And this is control mice, and this is inhibitor-treated mice. And the mean linear intercept, an indicator of lung airspace enlargement, decrease in inhibitor-treated mice. It is known that there are two types of macrophages in the lung: resident alveolar macrophages, AM, and interstitial macrophages, IM. AM originated from yolk-sac-derived EMPs and remain viable for a long time. On the other hand, IM originate from bone marrow-derived monocytes. And MMP12 is an elastolytic protease, also known as macrophage elastase. Previous studies in human and mice have shown that MMP12 degrades elastin in the alveolar wall and develop emphysema. It remains to be investigated what type of cells, AM or IM, produce MMP12 in the lung. Next, let me talk about monocyte-derived interstitial macrophages as the main source of MMP12 in the lung. This figure shows the gene expression of MMP12 in various cell types in the lung: neutrophil, eosinophils, basophil, mast cell, and monomac, T cell, B cell, and CD45 negative cells. Monocyte macrophages showed a higher expression of MMP12 compared to other cell type analyzed. And these monomacrophages infiltrated into the lung at around day 5. It was correlated with gene expression of MMP12. And to further characterize MMP12 expressing monocytes macrophages, we subdivided this population to monocytes, interstitial macrophages, and alveolar macrophages. Absolute number of monocytes increased at day 1, and IM increased at day 5, but had little effect on alveolar macrophages. Among these populations, IMs expressed a higher level of MMP12 than AMs and monocytes. These results indicated the importance of MMP12 expressing IMs in the development of emphysema. IMs are supposed to derive from blood circulating monocytes, which depend on CCR2 for migration into tissues. To determine the role of IMs in the development of emphysema, we utilize Seizure-2 knockout mice, which exhibit defective monocyte recruitment during immune responses. Monocyte macrophage populations are subdivided by surface marker of CD11B and F480 expression. In day 5, this population is IM, and this population is monocytes. The infiltration of monocytes and IMs into the lung was almost completely abolished in Seizure-2 knockout mice. Please see here. And while alveolar macrophage number was comparable between two mice. Furthermore, Seizure-2 knockout mice failed to exhibit an airspace enlargement. This is wild type, and this is Seizure-2 knockout mice. And MMP12 expression of whole lung was reduced in Seizure-2 knockout mice. And next, we performed adaptive transfer of bone marrow derived monocytes to Seizure-2 knockout mice. When monocytes were isolated from wild-type mice and intravenously transferred to Seizure-2 deficient mice before elastic installation, and they migrated to the lung and then here and differentiated it into the interstitial macrophages. In contrast, monocytes derived from Seizure-2 knockout mice failed to migrate to the lung. Importantly, adaptive transfer of wild-type but not Seizure-2 deficient monocytes reconstitute emphysema in CCR2 deficient mice. This result clearly demonstrated that monocytes recruited to the lung can differentiate into IMs but rarely AMs and contribute to the development of emphysema. To further characterize MMP12 expressing IMs, we analyze the expression of M2 markers in AMs and IMs. IMs exhibit M2-like phenotype, high expression of Arginase-1 and FIZ-1, which prompted us to investigate the role of IL-4. And gene expression of IL-4 was significantly upregulated at day 5. And IL-4 knockout mice failed to show airspace enlargement here. And the infiltration of IMs into the lung was abolished in IL-4 knockout mice, while the number of alveolar macrophages and monocytes was comparable. Moreover, MMP12 expression was decreased in IL-4 knockout mice. This result indicates that IL-4 is important for the MMP12 expression in IMs and the development of emphysema. In addition, we discovered that basophils showed a prominent expression of IL-4 in the lung here. And after illustrated installation, basophils accumulate in the lung with a peak on day 5. To clarify the importance of basophils in this model, we performed the depletion basophil using MCP8 DTR mice, transgenic mice. Diphtheria toxin mediated basophil ablation before the elastase administration ameliorates the development of emphysema. And the infiltration of IMs into the lung was significantly decreased in basophil depleted mice here, while AM number and monocytes number were comparable. Moreover, IL-4 expression and MMP12 expression of whole lung was reduced in the basophil depleted group, indicating the importance of basophil-derived IL-4 in the expression of MMP12. To directly assess the role of basophil-derived IL-4 in emphysema formation, we established MCP8 ICRE IL-4 flux device, but control mice is here, and this is mice deficient for IL-4 only in basophils. And these mice are resistant to emphysema formation, and which might show impaired IM generation and reduced expression of MMP12 in the lung. These results clearly demonstrated that basophil-derived IL-4 plays a crucial role in the generation of MMP12 producing IMs and development of emphysema. This is a report from Dr. Jordan, London University, show basophils in lung tissue of COPD patients. Lung resection samples were obtained from patients with mild to severe COPD and control undergoing surgery of the delineated tumors. For patients with very severe COPD, lung tissue was obtained from explanted lung after transplantation surgery. Immunohistochemistry revealed scattered bronchial eosinophils and here basophils in most patients. Eosinophils and basophils were present in all anatomical compartments of COPD-affected lung and increased significantly in very severe COPD, like called 4. This is the last slide. I'd like to summarize the main findings of our study in this schema. Blood circulating monocytes were infiltrated into the lung with CCL-dependent, two-dependent manner. Then basophil-derived IL-4 here play a crucial role in the generation of MMP12 expressing IMs. Finally, MMP12 expressing IMs leads the development of pulmonary emphysema and pathogenesis of COPD. That's all. Thank you for listening.